iPS cell. Usually we believe that the stem cells always can be differentiated to more specified cells. This is very similar to aging process. So cells can be always being aged further and further. Never thought it can come back to be younger. However, if the somatic cell's nucleus, if it is transferred to oocyte, then the new oocyte can generate the embryonic stem cell. That implies that maybe ES cell and oocyte, they may have some kind of pluripotency related factors. If we can utilize these factors, maybe we can change the somatic cell into pluripotent cells. This person, Shinya Yamanaka, who is the professor in Kyoto University in Japan, and he realized this crazy idea and then published the historical paper. For his work, he analyzed the gene expression of different type of cells, and then in this the graph, the red color is a highly expressed gene, and green color is a low expressed gene. And comparing to other cells, ES cells, they have some class of genes. They are much more expressed than regular cells. And he selected 24 candidates, which is overexpressed in embryonic stem cells. If this is your project, how would you design the experiment? How would you identify which factor is really important to maintain and to induce pluripotency? Before asking too many questions, he decided to express the 24 factors together in somatic cells and check whether some of those cells can change its fate into embryonic stem cell-like cells. But there was some problems. So one problem is the iPS generation frequency is very, very low. The other problem is the traditional staining method to confirm whether those cells are iPS or not, that requires cell fixation. Means you need to kill the cell and then confirm whether those cells are pluripotent or not. To overcome this problem, he chose FBX15 promoter and then reporter gene together. And FBX15 is an important factor for maintaining pluripotency. So if the cells are not pluripotent, then FBX15 is methylated and it does not make the reporter. But if we change the fate into pluripotent cell, then FBX15 is demethylated and then reporter can be generated. This reporter provides the resistance to antibiotics. Now, if you add this antibiotic to the cell culture, only pluripotent cells can survive. So when all 24 factors are present, then the survived cells, by overcoming the toxicity of G418, then these cells can be candidate of embryonic stem cell, and they chose the similar morphology at the colony to ES cells. For this experiment, they used all 24 factors to express and then confirmed the 24 factor can generate ES-like other cells. And then the next step is withdraw one by one of those factors. And if those factor is not important, then the efficiency may be similar to 24 factor. If the factor is very important, then maybe they can show much smaller the efficiency. Then the 10 best the factors are selected for the first round, and utilizing those 10, and again, the other factors are removed one by one, and finally, 4 factors are selected, and these 4 factors are OCT4 and SOX2, KLF4, and CMYC. And these 4 factors now we call Yamanaka factors. These 4 factors were tested again, And when out of these four, one factor is removed, then efficiency dropped a lot. But comparing to 10 other factors, this four factor is almost comparable. So that means maybe this four factor is enough. And maybe the other factors may interfere over the ES generations. So this four factor was finalized as key factors. When the morphology is compared, and original ES and starting point of MEF cell is very different, but they changed their fate ES-like, and the smooth ES-like colonies are formed. But some cases, the colonies is not as smooth as a regular ES, so this can be discarded. So selected, these are the new cell lines. They can be candidate of ES cells. When the gene expression pattern was compared using DNA or the methylation pattern, FBX15 is fully demethylated in ES, and MEF has a highly methylated pattern. Also, Oct4 nanoc, they have a highly methylated manner, it shows. Now, 10-factor based iPS, it has a lot of those methylation was removed, and 4-factor is a little less than 10-factor, but still it shows the quite strong embryonic stem cell property. So this result showed that the first example of possible way to make embryonic stem cell from somatic cells. So even though this is not perfect example, but this is a historical experiment. That's why this result awarded the Nobel Prize to Professor Gordon and Professor Yamanaka. So now we have the tool to make embryonic stem cell-like cells from any type of cell from our body. Now, if you think about the strategy to make the iPS, the original experiment to increase the efficiency, the retrovirus has been used. But the problem is that these are retroviruses. They can insert this external gene into our genomic structure, so it can be potentially dangerous. Later, this method was replaced by DNA virus, which does not insert this gene into the genome, but efficiency was lower. To make it even less impactful to the genetic changes, so plasmid also has been tested, even without any genes, just a recombinant protein. Purified protein was also used to demonstrate the iPS generation is possible. But still, the efficiency was not as good as original retrovirus method. Not only genes or proteins, small molecules were also tested whether it can replace those genetic vector or protein the functions. So some molecules were discovered, and very interestingly, but not surprisingly, those compounds are mainly epigenetic modifiers. So as you can see here, the compounds effect can convert the status of the methylated state of our cells into demethylated state of cells. Eventually, there was one paper came out, and they demonstrated that only utilizing the chemicals without any genetic material, and still they could induce pluripotent stem cells. So this method they call chemically induced the chemically induced pluripotent stem cell method. In this paper, they demonstrated that only utilizing four chemical molecules without any genetic material or proteins, still they could develop very high quality of ES-like cells. So see iPS here, and this is ESC, and the quality of the cells is much better than the original Yamanaka's other paper examples.